Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Advances in Precision Motion Imaging Clinical Trial Endpoints Collection for Parkinson's Disease. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, please contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario delivers the leading endpoint technology solutions for clinical trials. Through experience gained from over 19,000 clinical trials delivered in support of 870 regulatory approvals, Clario fuses scientific expertise and global scale into the broadest endpoint technology platform to enable pharmaceutical, biotech, and medical device partners to transform lives. Through Trial Anywhere, Clario has mastered the ability to generate rich evidence across all trial models, decentralized, hybrid, and site-based. And with 30 facilities in 9 countries across North America, Europe, and Asia Pacific, Clario's global team of science, technology, and operational experts have been delivering the richest clinical evidence for nearly 50 years. Now, I'd like to present today's agenda for the event. Today, we'll be hearing from Dr. Diane Stevenson from the Critical Path Institute, a neuroscientist by training, combines 30 years of experience in academic neuroscience and drug discovery. And from Clario, we have Dr. Kristen Sawalski, Director of Clinical Science and Consulting, who leads the Scientific Consulting Services team for Precision Motion. Dr. Faye Horak, Chief Scientist Consulting, and an internationally recognized physical therapist, neuroscientist, and author on all aspects of digital outcome measures related to mobility. We also have Dr. Martina Mancini, expert consultant, who studies how to improve everyday functional mobility and prevent falls with rehabilitation interventions. And Dr. Madhura Ingle-Hallecker, MRI scientist, neuromedical affairs, an expert in MR neuroimage acquisition, processing, and analysis. So now, without further ado, though, I'd like to hand the mic over to our first speaker today, Dr. Diane Stevenson. You may begin when you're ready.
Good morning, good afternoon, everyone. So great to be part of this exciting webinar today. And I'm going to dive right in and first tell everyone before we start that the disease of Parkinson's that we talk about today is so important in terms of unmet needs. This is the fastest growing brain disorders for reasons we're not really understanding yet. But it's very clear that there's a rich pipeline of therapeutic candidates in development, which is so exciting. Yet the urgent needs are around early intervention when promises of therapies can halt or even reverse disease progression. But we urgently need tools to tell us what measures we need to look at to identify as early in the disease and to track outcomes. And this is the focus of today's discussion. So we'll dive right in. I'm happy to represent Critical Path Institute, which has more than 25 different pre-competitive consortia. The one I'm going to talk about today is the Critical Path for Parkinson's Consortium. Next slide. And I've spent my entire career working on Parkinson's disease, primarily in industry, but when I came to Critical Path Institute, it became very clear that we cannot tackle the challenges for a disease like Parkinson's with even one organization. So I'm very proud today to highlight the work of the Critical Path for Parkinson's digital drug development tool team, which was launched just a few years ago. But even before we launched this digital project, Critical Path for Parkinson's has had success in neuroimaging. So it's very exciting for me to join today to highlight the work that transcends not just one tool, but to utilize multimodal imaging and digital tools for identifying the most promising candidates for trials. This is a very exciting project, very fast-paced, and we're making incredible progress because we have a unique forum to bring together industry, academic leaders, patients living with the disease, patient advocacy organization, and world leaders around the world to not only just talk about what's important, but to share data, share costs, and share risks together. And what's unique about this forum is that we have the regulatory agencies as part of our teams. We're grateful to the support from the US FDA and the EMA in helping shape what we can do together as a collective group. So the project I'm going to share with you today really, again, started because industry realized this was too difficult to do on their own. Next slide, please. And the big challenging question is, we know that to get these important treatments to the finish line, we need to intervene early. But it's incredibly difficult to identify people very early in this disease because there's not a lot of functional impairment. So how can these exciting new digital tools help identify people as early as possible? And what kind of measures are going to be useful to try to understand how to bring forward novel treatments at a time where little functional impairment occurs? So I want to thank the trailblazing work of our colleagues and partners at University of Rochester, Ray Dorsey, Jamie Adams and their team, who partnered with Biogen and Takeda years ago to design a very exciting seminal study that is a non-interventional observational study that I'm going to tell you about today. And this study started with just a couple of partners working together, but thanks to their generous sharing of a consortia-based approach, we've made incredible progress in addressing many of the gaps. Next slide. And this technology platform is really built on, a custom-developed iPhone app along with APDM Opal sensors that have been used for many years in Parkinson's, and you'll hear more about that today. But what was so exciting about our approach is, at Critical Path Institute, we're very known for our core competencies in bringing data together, usually after a study reads out. But this was the first time we ever went to the regulatory agencies to address and talk through a complex study design before the study ever started. And we utilized two forums to engage with the regulatory agencies. One is the Critical Path Innovation meetings with FDA, and a similar type of venue, the Innovative Task Force meeting forum with the EMA. These forums were not even in place 5 years ago, but they're designed to go forward to the agencies on a challenging issue that's not related to a single drug product or a therapeutic candidate itself, but what are these cross-cutting questions? And we had only 90 minutes, this was before COVID, in-person meetings, but boy, did we learn a lot. And we heard feedback that I'll just share with you at a high level, and we're very engaged on and dedicated to sharing these learnings, because the learnings we heard about this one single study are cross-cutting for all diseases. Next slide, I'll just share with you some high-level learnings that we heard and we're putting in place from these important regulatory meetings. The first thing we heard was a lot of issues around technical challenges. We really need to attend to quality of the data, rigorous collection of the data, standardization, and no black box algorithms. This is something the agency is seeing coming to them every day, and there are challenges that are not being easily addressed by one group. It's really important that we understand that Parkinson's disease is not just a classic motor dysfunction disorder that has been traditionally looked at for many, many years, that one needs to look at non-motor manifestations, which oftentimes are the most disabling thing to patients. And most importantly, we need to understand that these are not just fancy tools and technologies. We need to link what we're measuring with people living with the disease and how those digital tools link to what's important to patients in their daily lives. The main things we heard from them was very loud and clear that you cannot do a complex study like this in early disease without looking at healthy age controls. And then it's also to think about the application beyond an endpoint and think about how these sensitive tools can be used to identify people as early as possible. Again, this was before COVID, and here we are in this realm where we can go out and design these type of decentralized trials. Next slide. So we acted on the recommendations from the FDA in big ways. This meeting took place in 2019, and we've done a phenomenal job as a consortium with all the stakeholders in place, including people living with Parkinson's, to address these gaps. A couple of examples are shown here of publications that came out on addressing gaps in data standardization. I want to acknowledge Faye Horick, who's going to talk later, who led a whole other parallel effort on common data elements now that are now in place for us to collect data in the future in a much more rigorous, standardized way using digital technologies, and also try to identify sources of variability up front. Next slide. So what's so exciting is, again, we went to the agencies before the study started, and here we are. Now the fun part happens. The data is evolving, and we're really excited because, again, we implemented a normal control group. In fact, we put that study in place based on the regulatory agencies, and we had a parallel control group be co-funded by the consortium. And we're seeing some very exciting results. This is just an example of one of the symptom domains that showed significant reduction in arm swing on the affected side in the baseline results. This is just an example of one of many 5 different symptom domains at baseline that showed deficits, and we're now evaluating the results over the 1-year duration of the time. Important to note, at baseline, these patients did not have any dopamine medications, and then halfway through, half of the patients went on symptomatic meds. So lots of exciting results to analyze. And next slide. And we also are implementing a study that really came from the recommendations from our partners at the FDA on a qualitative study to link what's important to patients that are being evaluated with these digital platforms. So lastly, I'll say that we've learned a lot over the short few years since we started this project, but most importantly, we know that we're really in an exciting time and we have all the stakeholders at the table, but what we really need for success is much more attention to data standardization. Data sharing is the biggest gap. You know, for us to learn together, even if things don't work well, we need to share that knowledge to avoid duplication of effort and to make sure that people living with the disease are front and center to everything we do. So I'll pause there and just kind of hopefully give you this snapshot of the importance of working together. And I'm really excited to turn it over to my colleague, Kristen Zalewski, who's been part of the Critical Path for Parkinson's Consortium. And I'm excited to hear about the next presentation today. Thank you.
Thanks so much, Diane. Hi, everyone. So now I'm going to transition into looking at some overall considerations when selecting digital endpoints for your studies. So, you know, with selecting digital endpoints, sometimes this can be overwhelming, right? It's a challenge because there are a broad range of endpoints available, right? So how do we really narrow that down into selecting the specific digital endpoints that are most suitable for your unique clinical trial in Parkinson's disease, right? So one of the first considerations is, are the endpoints sensitive and specific for PD, right? Can these endpoints differentiate PD from healthy age-matched controls? And also, can they differentiate between PD and other indications? Another thing that needs to be taken into account is what stage of disease are the participants of a trial in, right? Are they in the prodromal phase, mild, moderate, or severe disease stage, right? So symptoms arise, you know, at different points of time. They don't all arise together, and they certainly don't progress at the same rates. That is definitely something to take into consideration when designing your trial with these endpoints. Additionally, endpoints, they should really reveal more insight versus some of the standard clinical assessments, right? Providing an electronic way to capture those assessments is beneficial. However, the main benefit of adding digital health tools is to provide more data and objective measures beyond what the clinical scales provide. And perhaps the most important bullet point here is that these endpoints must be meaningful to patient function, right? Everything we do all together collectively, the whole point of it is to provide treatments to help patients more effectively and more efficiently. Some other key considerations, we really need to understand the pathophysiology of the disease and understand the intent of the intervention and what particular part of that pathophysiology is it targeting, because that's really going to affect what endpoints you're going to want to be selecting. All right, and so moving into the next slide. So overall, what do we see in Parkinson's disease? How is this different from age-matched controls? So the first image that you'll see is of a person mimicking Parkinson's disease. Some of you may be familiar with this gentleman. But overall, this is a hypokinetic movement disorder based off of, the results of neurodegeneration. So those classic motor symptoms that we see are that forward-leaning posture, right? Difficulties with balance. We see a reduction in arm swing, and we see impaired postural stability and gait, right? We see smaller and slower steps, and that eventually turns into shuffling, right? So these endpoints are very important to capture, and not only capture what we're seeing, but to also capture in an objective manner those fine changes over time that we can't just see with the human eye, right? And so these endpoints are going to differ compared to, say, ataxia, which will be the next picture. Yep. So ataxia is going to be a different type of disorder, right? It's a cerebellar disorder, so we see impaired coordination and that staggering gait. So we're going to see more variability endpoints and larger movements in that case versus that hypokinetic disorder in Parkinson's disease. And also compared to, say, MS, right? That's a disorder in which involves demyelination and impairs the signal from the brain to the body, which you'll see in the next picture. So that, it requires a different set of endpoints. And if we can move into the next slide. So in the next slide, while we know what these endpoints are, we can kind of visually see some of them. We really need to assess with the patients which of these symptoms matter to them the most, right? So here we see a lovely survey done more recently by Moon and colleagues, in which 28 patients with Parkinson's disease were interviewed. And at the time of survey, they had answered questions around what symptoms they were experiencing at the time of survey, which is A, which of those symptoms are being insufficiently managed, which is B, and then on top of that, they asked the participants what they felt the research should be focused on. So you'll notice a trend here and see balance at the top, right? Balance remains at the top for all of those questions. And even though at the time of the survey, gait wasn't one of those symptoms they felt they were currently experiencing, the message was that it still is one of those top items that research should focus on. So in addition to surveys and really getting the voice of the patient, we need to understand how these endpoints relate to activities of daily living and overall quality of life. So, the classic PDQ-39 questionnaire is important to capture in trials, along with these digital tools to relate this type of data. So next slide, please. All right, so knowing the types of endpoints that we have to capture at these fine levels and what matters most to patients, we have to question, are the gold standard scales good enough, right? So the UPDRS and the HONE and YAR have been around for a long time. They're very important scales. However, they do have limitations, right? They are subjective in nature. They require skilled practitioners to perform them effectively. And they're on scales of 0 to 4 or 1 to 5. Right, so some of these limitations can, affect inter-rater reliability. And then additionally, they're not sensitive to the smaller but important changes between these different stages that are in need of capture. Okay, so how do we address these gaps? So on the next slide, you'll see that we need to start building these digital health tools into protocols to supplement those classic standard scales. Okay, so here we have the example today of our technology here at Clario, the Opal sensor technology device, right? So these sensors are placed in various locations of the body to instrument common motor assessments around gait and balance and provide objective, accurate, and reliable data in a way that reduces the burden on sites because of standard tests and sets of instructions so that it's consistent in between patient visits and across all site visits. And this technology can now be deployed both in the clinic for these active assessments and remotely as well, right? So it's the same basic technology, but through virtual applications, we can have patients capture these active assessments in the home, allowing more frequent data collection and less burden to the patients who are farther away from those clinical sites. So in the next slide, Now we're going to kind of transition and dive a little bit deeper into the particular areas in the tests that we instrument with these sensors to provide richer data, right? So these are really across three main domains of mobility, looking overall at gait, of balance, and then other tests of mobility that kind of target those postural transitions. So now I'm going to pass it along to Dr. Faye Horek to go further into endpoint collection in gait.
Thanks, Christian. Next slide, please. I'd like to talk to you why I think that gait is one of the most important outcome measures for clinical trials for people with Parkinson's disease. For one thing, the quality of how they walk influences whether they're going to fall or not in the future. And they fall 5 times more than people without Parkinson's disease. So it is a major and a meaningful health outcome for people with Parkinson's. In fact, there's measures of gait that can help predict falls, and I'll tell you a little about those. Also, quality of walking affects their ability to get around daily life and take care of themselves. And the other thing about gait is it's quite stereotyped. It's like a fingerprint. And so each of us walks a certain way very reliably day to day. And we know that it gets diagnosed, Parkinson's disease, based on how they walk, like the stiffness, axial stiffness of their trunk and the short strides and asymmetrical small arm swing. So it's very specific for Parkinson's disease too. And I'll be showing you how gait has different components, and some of the components might be more important for certain therapeutic targets than others. And it's very responsive to intervention. There's studies showing that it's responsive to levodopa. Gait is responsive to deep brain stimulation, but not all the subcomponents. Only certain ones are affected by different interventions. And it's sensitive to very early disease. It's one of the very first motor signs that you can quantitatively measure, even in people who have genetic predisposition for getting Parkinson's. Before you have any positive neurological signs, you can see and quantify these measures using body-worn sensors. Next slide, please. So in the next slide, I want to show you the different subcomponents of gait. If you use a stopwatch to measure walking, like for the get up and go test or 25-foot walk, you only are looking at the pace of gait. But when you use body-worn sensors, you get hundreds of measures. And many studies have now done principal component analysis and factor analysis to subgroup these measures to those that are relatively independent from each other. And they found that gait is really composed of quite a few different control systems. And the pace of gait, which is like the stride length and gait velocity, is one thing, and it's responsive to levodopa. But the timing of gait, such as the time you spend with both feet on the ground, which is called double support time, that is known to relate to fall risk, and yet it's not affected by levodopa. Gait variability also has measures related to fall risk, like stride time variability. And people with Parkinson's disease show more stride time variability. Asymmetry, as you saw, asymmetry of arm swing is very early in the disease, but not later. And stability of the upper trunk is affected by Parkinson's because they're stiff. In fact, they move their trunk less than they walk, whereas people with ataxia move their trunk too much when they walk. And turning is a special problem for people with Parkinson's. Okay, next slide. So I did a study, here's a goal, in which we wanted to understand how did levodopa affect all these different subcomponents of gait. So and also we included in here postural sway and standing, which Martina Mancini will talk about after me. So each color here represents a different component of gait and balance. And all the bars to the right show an improvement with levodopa in these over 100 people with Parkinson's disease who were measured before they took their medication and an hour after. And in light blue, you see upper body measures that improved a lot with levodopa, like arm swing, range of motion, and trunk mobility. And also pace measures like stride velocity and stride length. However, if you go down the graph, at the very bottom, you see that postural sway and standing actually got worse. So they had more sway and more imbalance. And if you look at the purple in the middle, you see that things like double support time, which is important for fall risk, doesn't improve or get worse. So if you have a target if you want to target something that is complementary to levodopa, you might want to target something here that levodopa doesn't help. But next slide, please. Another piece of evidence that all of these subcomponents of gait are really relatively, they represent different neural control systems, are imaging studies that are coming out. There's functional and structural imaging that supports this. And this is a brand new study I saw from Lynn Rochester's group, looking at PET scans where they're looking at metabolic networks, and found that there was a different network for pace of gait on the left, which involves the dorsolateral prefrontal cortex and SMA and the ventrolateral nucleus of the thalamus and other areas, whereas the temporal variability measures are very different parts of the cortex are involved, like the sensory motor cortex and the superior parietal cortex. So there's more and more evidence that gait is not just gait speed, and we need to be more specific depending on the question we're asking and the kinds of therapeutic intervention we have on what aspect of gait we think we can improve. Thank you. Next slide. When you look at the hundreds of measures and you say which measures are most sensitive and specific to Parkinson's disease, we've done this by including studies with just as many age match and gender match control subjects as people with Parkinson's, and found that one of the most sensitive measures is the angle of the foot at heel strike. This represents shuffling of gait. And we've found that this very sensitive measure is also correlated with measures to what patients care about. So we gave them the PDQ-39 on the right, and you could see foot strike angle is related to that. And on the left, the MDS UPDRS Part 2, which is the patient-reported quality of life measure, and that's also related to that. So these gait measures are related to what patients care about. Next slide. Another very sensitive measure and specific for Parkinson's disease is turning. Turn velocity is slower in people with Parkinson's disease, and you could see it was also correlated with the neurologist's judgment of the severity of the disease. So on the left is the correlation between turn velocity and the UPDRS total score, and on the right is the motor part of the UPDRS, the part 3. And turn velocity gets slower and slower as these neurological evaluations of severity of the disease gets worse. Next slide. So nice thing about turning is that it's abnormal when people are just diagnosed with Parkinson's disease. So here we did a study where on the left you see the traditional timed up and go test. It's a test where you stand up from a chair, you walk 7 meters, turn around and come back and sit down. And you can see the group with Parkinson's disease was not different than the control group. So they're very mild, newly diagnosed, not taking any medication, and yet their turn velocity was slow. And when we followed those people for 18 months, every 6 months, you could see that turn velocity started to slowly get, or turn duration got longer and longer, which means the velocity is slowed down with the disease. So we're hopeful, and in fact, we're starting a new study with over 100 people with Parkinson's newly diagnosed. And our hypothesis is that turning will be a sensitive measure of early progression of Parkinson's. Next slide. So turning here, you see a person's turning in their home during daily life. There's a Fitbit, there's a camera on their belt facing the ground so you can see their feet. And turning is important because you use it in most of your daily activities, and it's very hard for it to control. In fact, it's one of the sources of freezing of gait. If you want a person to see it freezing, ask a person with Parkinson's disease to turn. Also, if you turn, it's more likely that you'll fall during turning because it's a very dynamically unstable task. And if you do fall when you're turning, you hit your hip and you often fracture it. So it represents not only walking, but also dynamic balance. And it's easy to measure with a 2-minute walk test because we have a lot of 180 degree turns there to analyze. And I'm hopeful that turning will be even more useful than straight walking for people with Parkinson's disease. Okay, thank you for your attention. And now another aspect of balance control. Dr. Martina Mancini will be telling you about how balance is affected in Parkinson's and how we can measure it with body-worn sensors.
Thank you, Faye. And thank you everybody for attending. It's a pleasure to be here. Next slide. As Faye mentioned, I will be talking about balance. And as we saw, the aspect that Faye talked about were mainly related to gait. However, even when gait is improving or early on, when gait is still similar to healthy controls, there are aspects of balance that can already be impaired. How balance is measured from a clinical point of view, and you can see in the video that has been played, is by the pull test. The pull test consists in the administrator giving a tug to the shoulder at the patient. And as you can see in this example, we have an ONAR, for example, of 1 or 2 in an early person with Parkinson's disease, where there is only one step. And we can say that clinically, a clinician would say that there is no postural instability. However, there are aspects of balance that are very subtle and that cannot be seen by a naked eye, but we can still measure with inertial sensor. Next slide. So balance and mobility and gait has several different aspects or domains. Gait that has its own components, and Dr. Orak just talked about it. And then the pull test that we can see is actually on the fourth block, is assessing responses or automatic postural responses on how you are recovering from a perturbation. Those aspects tend to be also visually apparent, such as slow gait, or such as if a person can't recover from a perturbation and needs some help not to fall. However, there are 2 other aspects of balance: standing in postural sway, which basically is a condition in which we are supposedly standing still with our arms on the side or down in different sensory conditions, with eyes open, eyes closed, or with and without a foam support. When we are standing and pretending to be very still, there are actually continuous adjustments forward, backward, and sideways to maintain our balance. Those adjustments, although cannot be seen visually, are measurable with inertial sensor and are measurable traditionally in gait analysis lab with big force plate. Another aspect similar to postural sway in terms of how it's measured are transition, and in particular case, the one that occur before taking the first step. So if you are standing and you have to get going, we have a preparation to start walking, consisting in weight shifting. Next slide. As I was just mentioning, postural sway has been measured traditionally with a force plate by looking at the characteristic of the center of pressure, which is the point of contact in between the feet and the force plate, reflecting the forces exchanged. However, years ago, we demonstrated that we can get a similar information by placing an accelerometer on the lower back, more or less at the center of mass level. And next slide. And that acceleration of the center of mass is directly correlated with the center of pressure excursion during postural sway in this semi-static condition. On the figure on the right, we can see the statokinesigram, which is basically the acceleration in the anteroposterior versus the mediolateral direction in a healthy control. As you see, it's consisting of continuous adjustment forward, backward, and sideways to maintain balance. In a person with Parkinson's disease, you can see how these oscillations are larger, especially in the mediolateral direction. From these acceleration traces, we can characterize the sway area, the amount of movement, sway velocity, the jerkiness, frequency domain, and some of those domains have been related to fall risk in both elderly control, and people with Parkinson's disease. Next slide. And if you remember the first video that I showed where the individual recovered very well from the pull test, and that was the novo untreated person with Parkinson's disease, for example, as similar to turning, when gait speed is still normal, actually postural sway is impaired already. As you can notice by measuring postural sway with the accelerometer on the lower back, we have an example of an healthy control and an individual with untreated Parkinson's disease, showing increased excursion in the mediolateral direction, increased jerkiness and frequency. So we can say that those digital markers of postural sway are sensitive to early stage of the disease. And there are papers also showing impairments in the prodromal phase of Parkinson's disease or postural sway. Next slide. Regarding sensitivity to and responsiveness to medication and levodopa, in the figure we have some representative examples of two individuals, one who will undergo deep brain stimulation in the globo pallidus internus, and one who will undergo deep brain stimulation in the nucleus subthalamicus. And as Dr. Orak mentioned before, while aspects of gait were improving with levodopa, who is the mainstream medication for Parkinson's disease, aspects of postural sway are actually worsening with levodopa. We can see a larger sway and faster velocity, posing an individual maybe at higher risk for falls. And next, and this is work that is in progress. After the brain stimulation, we see that while the brain stimulation in the globus pallidus internus is effective in reducing postural sway, the brain stimulation in the nucleus subthalamicus is not. There is no change from before and after. We can go to next slide. Another aspect of balance, which is not easily accessible by a naked eye, is step initiation in Parkinson's disease. Has been measured again with a force plate, and it consists on shifting the weight of the to move the center of mass forward for the first step. There is a characteristic pattern of the center of pressure, which consists in moving backward and sideways in order to propel the center of mass to take the first step. If we have an accelerometer on the lower, at the level of the center of mass, we can see exactly a mirror image of the center of pressure. And what we characterize actually the amplitude of the excursion of the center of pressure or the acceleration. And if we look at next slide, and this marker, again, in the same cohort of untreated PD that we investigated, we can see that while there is a large preparation for the first step in a healthy control, in untreated people with Parkinson's disease, that preparation is already compromised and much smaller. And that could be later on a marker of start hesitation. For example, people with freezing of gait sometimes hesitate to take the first step. And we know that people with freezing show a reduction in step initiation preparation. Next slide. In contrast to the aspect of postural sway, the aspect of preparation to take the first step are actually much improved with levodopa medication. As you can see, and here we have the curve indicating the magnitude of the preparation, and we look at the peak over here compared to baseline. This peak is much higher in the levodopa condition compared to the off condition. However, for both the brain stimulation in the GPI and STN, there is actually not a change after the brain stimulation, but a slight worsening. So this means that when we have a new drug, we want to consider a digital marker, more than one, a digital marker that are including different aspects of balance, because some of them are responding well, for example, to a certain medication, but others might be worsening. And that's also why maybe people with Parkinson's disease, also after deep brain stimulation, they are still falling, even if their gait speed sometimes is much improved. And next slide, and last slide. It's a further proof that these components of balance are also very different. We recently conducted a resting state functional connectivity study. And we found that the ventral attention and ventral multimodal resting state functional connectivity were predicting amplitude of postural sway. And this is the figure on the left. And the frontoparietal and ventral attention resting state functional connectivity predicted the preparation to step initiation. And these observations are consistent with the hypothesis that both standing and step initiation are cognitively controlled in people with Parkinson's disease and less automatically controlled. And the next slide, I'm actually gonna introduce Dr. Madhura Ingahalikar, who will talk more about the imaging components.
Hello everyone, my talk will be primarily focused on imaging endpoints in Parkinson's disease. Now from the earlier 4 talks, we have already seen how PD involves multiple circuits. Next slide please. Such as the motor circuit, the limbic circuit, as well as cognitive circuits as shown in this figure. Now, these circuits reveal a variety of anatomic and functional targets that are amenable to neuroimaging and biomarker discovery. Not only imaging can be used for patient recruitment and safety, but also as efficacy endpoints. And with this brief background, I am going to talk more about how imaging can be incorporated in PD clinical trials. Next slide, please. The first thing that strikes as the most relevant in PD is a DAT scan, which is a dopamine transporter SPECT scan, and is critical in eligibility assessment in PD trials. Now, abnormal uptake in the putamen or caudate is very consistent with the DAT deficit, which is typical of PD, and as shown in the figure here. So you can see that in the putamen or the caudate, the uptake is abnormal compared to a normal healthy control. Now going to the next slide, in terms of MRI, the eligibility and safety monitoring is very critical in PD, and it includes a standard clinical protocol, which includes the T2 imaging to detect white matter lesions, FLAIR, which is the fluid attenuation with inversion recovery, where the CSF is suppressed, for enhanced visualization of lesions, tumors, and edema, diffusion ADC maps, T2 star imaging to detect blood clots and microhemorrhages, and also a 3D T1, which is a 3D anatomy scan, which can also be used in efficacy for measurement of volumes in the basal ganglia, as well as gray matter regions of interest. Now, in this slide, another interesting MR modality that can be very well employed in PD trials is diffusion MRI. Now, this is very beautiful because what the easiest way to explain diffusion MRI can be considered as the Google map of the brain. So what you're trying to see is, are these beautiful white matter tracks that traverse and connect different regions of the brain. So that's why I'm calling it as a Google map of the brain. So what we are looking through diffusion is a macro model of this actual physical pathway. Now, diffusion MRI is actually sensitive to the diffusion of water, which is directional in the white matter because of its fiber-like structure. By mathematically modeling this as a tensor, we can track the white matter fibers as well as use anisotropy measures to understand the density and the integrity of white matter. Now, studies in PD have consistently demonstrated that white matter integrity in PD is much lower than in healthy controls, as shown in the chart here, the bar plot here. More than white matter analysis, by using better mathematical models, we can also extract something called as the free water. And the free water is nothing but the extracellular water contained, which is kind of an indication of neuroinflammation. Now, this is shown in the figure next here, and here you can see that from the studies, the free water content eventually increases as the disease, as the Parkinson's disease progresses. Now, this is not just in the basal ganglia, but also in areas like substantia nigra, where we can see a change, a significant change in the free water as the disease progresses. Next slide, please. Now, diffusion MRI, when acquired with higher angular resolution, we can use much better microstructural models that overcome the partial volume problems and can support improved anatomical white matter tracking, as well as provide more meaningful and precise white matter measures. Now, as shown here, higher order models can help traverse through the white matter crossings. So you can see in the first picture here, the crossings can be easily traversed through. And therefore, the tracking of fiber tracts such as the dentatorubro-thalamic tract, which is important in most of the Parkinsonian disorders, can be performed with such advanced diffusion MRI. Moreover, advanced diffusion matrix such as the apparent fiber density have shown significant longitudinal effects in Parkinson's disease, as shown in this figure here. Now, next slide, please. Now, peculiar to Parkinson's disease is something called as the neuromelanin sensitive imaging. Neuromelanin sensitive MRI can detect the content of neuromelanin, which is a product of dopamine metabolism that accumulates in the substantia nigra pars compacta. Now, given the neuromelanin ion complexes are paramagnetic in nature, these can be imaged through MRI. The neuromelanin images illustrate atrophied substantia nigra pars compacta in the patients with Parkinson's disease. Next slide, please. So we performed a study to see how the contrast changes between the substantia nigra pars compacta and the background. And we saw that the contrast eventually vanishes between the substantia nigra and the background as the patients become more severe in PD, as the disease progresses. So this is what we see. More than just finding the contrast ratio and understanding the area of substantia nigra pars compacta, this imaging is important because we can actually use it to find the region of interest, which is the substantia nigra pars compacta, which is hard to find on any other modality. And this can be mapped to other modalities such as diffusion MRI to find out, say, the free water changes in the substantia nigra. So this is why the neuromelanin imaging is very important in PD clinical trials. Going to the next slide. It is believed that the dopaminergic neuronal death in the substantia nigra pars compacta, mainly the nigrosome area, is followed by neuromelanin deep pigmentation, and it leaves behind iron that becomes visible on the MRI, making iron imaging another important modality in Parkinson's disease to map the nigrosome, mainly to map the nigrosome. Now, the good news is that although there are not too many studies on iron imaging in PD, the good news is that when you acquire neuromelanin MRI, you can also acquire iron imaging in the same acquisition protocol. So it's not something else, like you don't have to do it separately. You can acquire it in the same acquisition protocol itself. So that makes easier to acquire iron imaging as well. So it reduces the total scanning burden. And finally, on the next slide, I'm just going to talk about functional MRI. Now, I talked about diffusion and I said that that is the Google map of the brain because you have these pathways that connect A to B. But now with functional MRI, this is another type of a Google map, just that now you're not looking at the pathways, you're looking at the traffic patterns on the Google map. So you're looking at how the functional, when you are performing certain function, how the bolt signal varies. The bolt signal is the blood oxygen level dependent signal. So that is what you're trying to see. Now, in PD, we expect the frontostriatal connectivity to reduce and cerebral visuo-inferior parietal connectivity to increase. Also, with cognitive deficits, we expect to see reduced default mode and executive network connectivity. And also, I mean, earlier we saw like how Martina showed the functional connections. So these can be used in studies to find out how gait and balance and how these mechanisms are actually related to what networks in the brain. And that is what is very important. So that's why fMRI becomes one of the very crucial modalities for PD clinical trials. Going to the next slide. Now I'll just try to summarize the imaging part. Now imaging is an important tool in PD clinical trials and is required for eligibility, safety, and efficacy. Now integration across all the imaging endpoints is really crucial, and that will yield the most precise characterization for PD. Now I'll take one step further and say that integration across not just the imaging endpoints, but also with precision motion based endpoints, we can definitely have a much better characterization of the disorder. So that's it, and I will end my talk and hand it over to Christine to do the summary for all of us. Thank you.
Thank you, Madhura. All right, so before I go into the complete summary, on the next slide, I have to bring up and address the recent release of guidance, right? FDA guidance on the use of DHTs in clinical trials. So I've highlighted particular places in which there are recommendations that we've sort of spoken about a bit today. So one of those key recommendations is that endpoints need to be selected to provide a broader picture of how patients feel or function, right? So we really looked at that survey, and we want to make sure we're getting endpoints around things that patients are rating as really important to them in their daily lives. All right, and then DHTs can be used to administer some of these assessments electronically, right? And so much of the work that we spoke about, specifically within precision motion, are instrumenting electronic versions of those performance outcomes and providing further endpoints around those. As Dr. Stevenson mentioned, it's very important to engage early with regulatory agencies to discuss the use of DHTs in your trials. And if you hit next, we've got some more bullet points. Thank you. And then, you know, there's a summary of some of the main considerations, right? We really need to understand what the characteristics of the disease are that we are trying to capture. All right, and it needs to be specific to that indication. And then it really needs to be implemented into the design of the clinical investigation, right? Does it have in-clinic components? Does it have remote components? Is it active assessments? Is it passive assessments? Those types of things. When assessing the technology to capture these endpoints, right, it needs to be items around ease of use, right, for both site and for patients, sufficient battery and power life, data storage capacity, and transmission of data. These endpoints must be fit for purpose, right? What's the objective of the trial, and how do we capture those endpoints, and making sure it's a good match. Excuse me. And then with the tools, we want to make sure that they've been verified, right? Is that technology measuring what it says they're measuring? And then we want to make sure they're validated. Can those those digital endpoints be used in that particular indication? Right? And then usability. It's very helpful to have feedback from participants, from patients, on how the experience was with the technology. So it's important to look when assessing which tools to use to take all of these things into consideration. And then you can use these tools, again, to electronically capture established clinical endpoints, right? So in movement, we can do that with, say, gait speed or walk distance. But you know, the real opportunity here is to produce novel endpoints. Yep. And so, you know, and hopefully we gave some examples of that today, and how instrumenting some of those components that are captured generally in the UPDRS, like questions around balance and gait, by instrumenting those tests, we can provide novel endpoints to provide more evidence supporting those types of questions. All right, and then to summarize on the next slide, we really want to emphasize the importance of really everyone coming together, right? Aligning with your industry and regulatory colleagues to all achieve the same goal, which is really to help patients by advancing therapeutic interventions and their efficacy. Just bringing up some of those key considerations, right, that we've mentioned throughout the course of this presentation, and these are things that we are here to help you with. So we hope that today you've had a chance and have learned a little bit more about using precision motion and imaging endpoints to help generate that rich objective data in Parkinson's disease by providing measures that are sensitive, accurate, and reliable. And with that, we thank you all very much for your attention and for attending, and we are happy to answer any questions that you may have.
Well, thank you very much for that insightful presentation. I'd like at this point to invite our audience to continue sending their questions or comments right now using the questions window for the Q&A portion of this webinar. Now, I've already received some questions for you all, so we'll get ourselves started with those. The very first question I have here, I believe, should be directed towards Dr. Stevenson. It asks, are there case examples where harmonization and integration of data across distinct device platforms have been successfully executed to enable device agnostic strategies?
Yes, thank you so much, Ryan. That's a really great question that we are all looking for examples, right? I know of the European initiative Mobilized that is doing this with Gate, but the holy grail is really to not just look at one device opportunity, but to try to do this harmonization. We do this at Critical Path Institute with traditional measures across many different diseases. In this field, which changes very fast, this is an example where we actually look to you to come up with and help us address these questions, because this is going to be important rather than looking at just one example at a time. It's a really important gap for us going forward.
Excellent, thank you for that. Next, Dr. Horak, I have someone asking, why is turning so much more difficult to control than straight ahead gait? Sorry, Dr. Horak,
Sorry,
there you are.
I was muted, sorry. But yeah, turning is complicated because normally we start with our eyes, then our head, then our trunk, then our legs. So it's a complicated coordination task and a sensory task. And people with Parkinson's disease, they don't start with the eyes and then the head and the trunk. They turn all as a unit. And so they don't have good control of their body center mass. So it becomes very close to the limits of their foot, which means they're much more likely to fall sideways. So I think it's because it's such a complicated sensory motor task and such a difficult dynamic balance task.
Excellent, very good, thank you. I want to get to a question for Dr. Engel-Hallacher, asking, can you run these MRI acquisitions in typical clinical setup?
Hi, thanks Ryan for the question. Yes, I think most of the MRI acquisitions that you saw in my presentation today, at some level, yes, we can run it on most of the clinical sites, especially the safety and the safety protocol can be easily run anywhere. And for the advanced protocols, if they have the packages, yes, we can run it. And we have had very good output with with clinical trials and experience with most of the clinical trials where we can also run advanced protocols very easily. Thanks.
Very good, thank you very much for that. I'd like to get a question for Dr. Mancini. I have someone here asking, how long should a postural sway trial last and why?
That's a very good point. We do recommend at least 30 seconds, which is a compromise in between like a longer recording and something that is practical for clinical trials. We want 30 seconds to ensure reliability of the amplitude measure, for example. So that's what we recommend at the minimum.
Very good, thank you. Now, Dr. Sawalski, is there a risk of data loss by instrumented assessments with the Opal?
Thanks, Ryan. We've got sort of several backups for data with how we capture data with the Opal. So the Opal itself has inertial measurement units in it, and that data is stored, that raw data is stored on the sensors itself. But while these instrumented tasks are being performed, that data is also transmitted wirelessly to the computer live. So it's being backed up there, it's going right into the computer, that raw data, which is then run through algorithms. And so the process endpoints are also produced, and both sets of data, that raw data and those endpoints, are then backed up into the cloud. So there's really minimized any risk of data loss because of those three backups there.
Very good, excellent, thank you. Dr. Sawalski, I have another question here for you. Wondering, why wasn't sleep a suggestion for researchers to focus on when that was one of the things very high on the list of what was important for patients?
Sure, that's a great question. And first, sleep is essential, right? Sleep's very important. And I think Dr. Stevenson did highlight that it's very important to capture non-motor symptoms. And that is one of those items that can be captured either with clinical scales or with a digital sensor, right? In like actigraphy mode in sleep endpoints. So I think that is important. I think with the survey that I presented, what it really highlighted was how important it is to patients to be able to move around confidently, right? Sort of their functional mobility. And that's why we saw kind of more of those endpoints as recommendations for what researchers have focused on, right? Because if there's lack of balance, you know, either while standing still or moving around, there's fall risk. And so, you know, I believe that kind of highlighted the importance of that. And then, you know, we did focus on our presentation today, mostly on those precision motion endpoints and imaging. So I hope that answers the question.
It's wonderful. Thank you very much for that answer and for all of the answers. However, we have reached the end of the Q&A portion of the webinar today. Now, if we couldn't attend to your questions, though, the team at Clario will follow up with you. Or if you have further questions, you can visit clario.com/contact. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thanks for coming.
Thanks, everyone.